Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 13 from our Genetics Essentials Concepts and Connections 4th edition book. This chapter deals with gene mutations, transposable elements, and DNA repair. We're not going to be speaking too much on transposable elements, but we are going to be focusing on gene mutations and DNA repair. So what are mutations and why are they important to genetics? It says here something profound. It says mutations are the sustainer of life and the cause of great suffering. And I couldn't have put it better than what your book said there. The reason life exists today is because of genetic mutations in populations over time, which is essentially evolution. Populations have adapted in order to suit their environment, in order to persist and proliferate and carry on their existence over time. And mutation is important, and genetic diversity is important to the sustainability of life. However, at the same time, mutations are a cause of great suffering for populations as well. Think of genetic disorders, genetic diseases that you can inherit. Think of mutations that could lead to cancer, and what a big plight cancer is. So, this is what we're talking about when we talk about mutations being a cause of great suffering. Think of bacteria that mutate in order to become resistant to antibiotics. That causes suffering. Think of viruses that mutate and become even more virulent, even deadlier, right? Like the flu. The flu is constantly mutating and becoming worse, right? Even with COVID nowadays, one of the fears is that it could mutate and become worse, right? Mutation is the leading cause of cancer. You're changing DNA around. You're affecting important cell cycle regulatory genes, and those genes no longer function in the cell because the gene products, those proteins that are involved with the cell cycle. Remember we talked in the last chapter about how important the cell cycle is, and that if you escape the cell cycle, then a skin cell might divide too often, or lose its ability to regulate itself, and next thing you know, you have a clump of cells that's a tumor, and then that could metastasize, and there you go, there's cancer. So again, mutations so important to life. It's so important to increasing genetic variability, and in order to adapt and mutate and persist, but also a source of horrible suffering as well. So let's talk about some categories of mutations. Somatic mutations, this means mutations in your non-sex cells. Germ-line mutations, this means mutations that are occurring during meiosis in the gametes. So these would be inheritable mutations. Let's say a particular gene is mutated in the germ-line cells, in the testes, for instance. Well, then those mutations could get passed on to the sperm, and then those mutations could be stably inherited by the offspring, by your offspring. So your offspring would have that mutation throughout its body, right? Whereas in a somatic mutation, let's say a lung cell or a skin cell gets mutated, in that case, you're not going to pass on that mutation to your offspring. You're not going to pass that on. Now, that may cause cancer on you, but you're not going to pass on that, that mutation to your offspring. In order to pass on a mutation to your offspring, that mutation has to be germ-line, which means the sperm or the egg need to possess that mutation. And there's only two ways for that to happen. One is you were born with that mutation, so you're automatically going to pass on that mutation through your sperm because every cell in your body should have that mutation because you were born with that mutation. The other way is, another spontaneous mutation or a induced mutation by exposure to chemicals or something, but that specifically happened in your germ-line cells in the process of gametogenesis. So we're going to talk a little bit about genes and gene versus chromosomal mutations. So take a look here. Here's an example of what I was just talking about. A somatic mutation and its effects versus a germ-line mutation and its effects. Remember, in a somatic mutations occur in non-reproductive cells. So let's say this cell right here on this zebrafish. Well, then that cell will undergo mitosis and it might become a clump of cells, but that's, highly localized to this area. Okay, are passed to new cells through mitosis, creating a clone of the mutant cell, having the mutant gene, but it's restricted to this area, this patchy area on the zebrafish. And again, that could result in something like cancer or apoptosis of the cell that's mutated, but it's not going to be passed on to this fish offspring. However, if a mutation were to occur, let's say a spontaneous mutation were to occur right here in the gonads of the fish and its germ-line, then in the germ cells specifically, well, then in this case, you could pass on that mutation to the gametes, the sperm or the egg, and then you would find that you would either have mutant offspring or wild-type offspring, depending on whether or not this happened in one allele or both, but most likely if it's a spontaneous mutation, it occurred in one allele, not both. So let's talk a little bit about the types of gene mutations that can occur. So these are gene mutations. You could have base substitutions, you could have insertions and deletions, and you can have expanding nucleotide repeats. So let's talk about each. With base substitutions, you have what are known as either transitions or transversions. With a transition, a purine is replaced by a different purine. Pyrimidine is replaced by a different pyrimidine. So you're not changing what type of nucleotide. You're keeping one-ring nucleotides the same, or you're keeping two-ring nucleotides the same. But that's it. But in transversions, you're switching from a purine with two rings to a pyrimidine with one ring, or you're switching from a pyrimidine with one ring to a purine with two rings. In insertions and deletions, you can have, let's say you have a genetic code, right? A, G, T, G, C, A. You have a genetic code. What if you were to add a letter somewhere in the gene, right? Let's say you add a guanine to the gene somewhere. Well, that would shift your reading frame. Remember, because we learned in translation that when we were studying translation, we learned that starting at the start codon AUG, that sets the reading frame. So AUG is the first codon, then the ribosome will read the next three nucleotides, and then the ribosome will read the next three nucleotides. Does that make sense? That's your reading frame. Your reading frame is set by the start codon AUG. And then the ribosome knows to read every three nucleotides after that point, because every three nucleotides will correspond to the anticodon on the tRNA, and translation can persist. But what if you were to add a letter to the gene somewhere? What if you were to subtract a letter from the gene somewhere? Well, that would cause a frame shift mutation. This changes the reading frame, and usually everything after a reading frame mutation becomes gibberish. So you could even have in-frame insertions and deletions. What does that mean, in-frame? Well, what if you were to add 3 nucleotides or delete 3 nucleotides? That wouldn't affect your reading frame, would it? If you were to add 3 nucleotides or subtract 3 nucleotides or some multiple of, that shouldn't affect your reading frame. That won't shift the reading frame. That'll just introduce a whole new, maybe it'll introduce an amino acid or it'll subtract an amino acid. Does that make sense? Or some multiple. And what's a expanding nucleotide repeat? This is increase in the number of copies of a set of nucleotides. So we'll talk about that in a little bit. Let's first talk about gene mutations, specifically that have to do with the 3 basic types. The 3 basic types of gene mutations are, remember, substitutions, insertions, and deletions. Substitutions, insertions, and deletions. So this is your original gene sequence. You've got GGG, AGT, GTA, GAT, CGT. A substitution, now you're substituting this thymine and you're substituting a C in for that thymine. So now you've got a cytosine where you should have a thymine, you see? So would this be a transition or a transversion, do you think? If you're substituting a C instead of a T, what do you think? Well, isn't C a pyrimidine and T is a pyrimidine? So in that case, it would be a transition mutation. This would be an example of a transition mutation, and only one codon has changed. Let's look what happens in a frame shift. Remember, a frame shift mutation where I'm adding a letter. So I'm just going to add a T here, a thymine here. So now instead of saying GTA, it says GTTA, right? I've added a thymine, and remember what this does, it effectively shifts my codon sequence over, it shifts the reading frame. So now as the ribosome reads through this information, it'll read GGG, AGT, GTT. You see, so that everything after that frame shift mutation is now gibberish, because the genetic code should read GTA, GAT, and now it reads GTT, AGA. You see how all the codons are now incorrect, right? And the same would happen if you deleted a base. You see this? Instead of GTA, you cut out that T, and now it's GAG instead of GTA. Well, again, you're going to cause a frame shift, and that's going to cause gibberish for the rest of the gene. These are highly deleterious mutations. Frame shift mutations are highly deleterious. Now here's an example of transitions versus transversions. Remember, transitions, you're going from a purine to a purine. So you're going from an A to a G or a G to an A, basically a 2-ring structure to a 2-ring structure, or a pyrimidine to a pyrimidine. So this would be a T to a C to a T. If we were dealing with RNA, it would be a U to a C, or you would have U's instead of T's. Transversions are when you're the mutation, you're swapping a pyrimidine for a purine. This would be A to C, A to T, G to C, G to T, or a pyrimidine for a purine. Okay. So concept check number 1, time. Which of the following changes is a transition base substitution? Okay, which one's a transition? Remember what transition means? You don't change the identity of the purine or pyrimidine. It stays a purine, right? Or it stays a pyrimidine, right? Transition. The other one would be the transversion. So we're talking transition, staying a purine or staying a pyrimidine. So adenine, which is a purine, for thymine, that's a pyrimidine. Nope. Cytosine, which is a pyrimidine, for adenine, which is a purine. Nope. Guanine, which is a purine, for adenine, which is a purine. C. Yes, so it's C, because guanine has 2 rings, it's a purine, and adenine is a purine as well. It is a 2-ring structure. Okay, and the other thing that we were talking about was repeats. Let's go back to this. Remember expanding nucleotide repeats? This is increasing the number of copies of a set of nucleotides. Sometimes mutations can occur where you take repeats. Remember we talked about repeat sequences in the genome and how frequent they are? Well, some mutations can result in those repeats being copied even more, right? And that can cause these large segments of repeats to form. Fragile X syndrome is an example. Fragile X syndrome is associated with a characteristic constriction, this is called the fragile site, on the long arm of the X chromosome, and is caused by expanding nucleotide repeats. This is the most common cause of heritable intellectual disability. Interesting. Here are some more examples of those repeats, those expanding nucleotide repeats, and the diseases that are caused by these repeats. And this is a very important point up here. Increases in the number of nucleotide repeats, just in case you're wondering why it's so bad to have these repeats. Increases in the number of these repeats can produce disease symptoms in different ways, such as it could interfere with normal genes. Let's say the repeats happen and they disrupt a normal gene. They happen in the middle of a normal gene. They could affect normal gene expression. What if they disrupt an enhancer for a gene, or a promoter for a gene, or an insulator for a gene, right? And they can induce DNA methylation, and there's other techniques by which they can disrupt the, your genes as well. They could disrupt normal function or development of your cells as well. So these are examples. Famous examples are Huntington disease. You probably heard of Huntington disease, Fragile X disease. All right, so let's keep moving on. So what are the phenotypic effects of mutations? Now we know how we get mutations in the genes. What are the phenotypic effects of mutations? Well, you could have what's known as a forward mutation. This would be a mutation that, causes a mutant type phenotype. So you've got red eye flies, for instance, and now you've got a white eye fly mutant. Reverse mutation would be you go from a mutant back to wild type. Let's say whatever caused the forward mutation, what if you reverse it? What if it by chance got reversed in, the offspring of that fly? Well, then you would have what's known as a reverse mutant. Missense mutation, this is when the codon codes for a different amino acid. So the codon was mutated in such a way that it now codes for a different amino acid on that table. Remember the codon table. And this would be most likely to happen if you mutate the first or second positions of the codon, right? If you mutate the third position of the codon, you're most likely going to get a silent mutation where the codon becomes a synonymous codon. So like CCC becomes CCG, and most likely because that's the wobble position, the third position on the codon, you're going to get no change in the amino acid it codes for, and you're going to get no change in the protein. So silent mutations are an actual change in the DNA, but it doesn't change the amino acid sequence of the protein. Missense mutations are a change in the DNA that does change the amino acid sequence of the protein. Nonsense mutations, this would be if you had a mutation that resulted in a stop codon. So for example, UGA is a stop codon. UGA, what if a mutation went from CGA to UGA, right? CGA codes for an amino acid, but you mutate that to UGA, that's a stop codon. So now you've introduced a premature stop codon. A nonsense mutation is a mutation that results in a premature stop codon. And then a neutral mutation is kind of like the in-between a silent mutation and a missense mutation. Okay, think of neutral mutation as an in-between of silent and missense, because it is a missense mutation in that you change the amino acid, but you change it to a very similar amino acid, And because it's a very similar amino acid, what do I mean by similar amino acid? Maybe you change a non-polar amino acid to another non-polar amino acid, or you change it from an acidic amino acid to another acidic amino acid. Well, then, because it's so similar, the protein may fold just fine and function just fine, right? So that would be a neutral mutation, where it's actually a missense mutation, but not a deleterious missense mutation. So, for example, it would be a lot worse usually to have a acidic amino acid be mutated to a basic amino acid, right? Because now instead of a negative charge, you got a positive charge. So now the protein will unfold or something, right? That might unfold. But if you were to mutate an acidic amino acid for another acidic amino acid, well, then there's no real reason for a lot of repulsion to happen in the protein, and there's no real reason for it to unfold and stop its function, right? So think about it that way. There are such things as really bad missense mutations, and there are such things as meh missense mutations, and a neutral mutation is a meh missense mutation. Here's some more phenotypic effects of mutations. Loss of function mutations. This would be a mutation that results in a non-functional protein usually. Gain of function mutations. This would be a mutation that grants a function to a protein. Conditional mutation. This would be a mutation that only expresses a phenotype under certain circumstances. Do you guys remember the bunny rabbit I showed you? And the bunny rabbit had a black fur on its ears, nose, and tips of its paws, and its tail, only where it was cool. So that would be an example of a conditional mutation. That was a temperature sensitive mutation, right? The protein showed its mutant phenotype in only in warm parts of its body, in its belly and stuff, where it's warmer. That protein was kind of, it unfolds so it doesn't do its job to make pigment. But in the cooler parts, like in its ears, that protein folds properly and does its job properly. So that would be an example of a conditional mutation. And then you have lethal mutations. Remember agouti, fur color in mice is an example of a lethal mutation. If you have not agouti, I'm sorry, yellow fur color in mice, yellow Y, big Y. If you have two big Y in mice, that can result in a lethal phenotype, right? So you can have lethal mutations, a mutation that's actually dangerous to have. Okay, here you can see the examples I was just talking about: missense, nonsense, and silent. So here's what the sequence would say on a no mutation. The triplet codon reads AGT, right? And that the mRNA is UCA, right? Because the mRNA, the complement of A is U, the complement of G is C, and the complement of T is A. And UCA, if you were to go look at that 64 or 4 by 4 by 4 codon box, remember the codon cheat sheet that we had in the previous chapter? If you were to look at the codon table, you would see that UCA codes for serine, the amino acid serine. So this is what you would expect in the wild type protein. But in a missense mutation, you're going to change a letter. So let's say you change TCA to TTA. Well, now, AGT to AAT. Well, now it's going to be transcribed to UUA. And UUA, if you were to check out the codon table, it actually corresponds to the amino acid leucine. So instead of serine, you have leucine, which is not good, right? Here's a nonsense mutation. So remember, nonsense is where you change an amino acid like serine to a stop codon. How could you do that? Well, let's say AGT that coded for serine is now ATT, and ATT corresponds to the stop codon UAA. So now you have a premature stop codon. So guess what? Now you have what's called truncated protein, which means you've only built part of your protein, and that protein is likely not going to function if you've only built part of it. Does that make sense? And then a silent mutation usually occurs in the wobble position. Remember the third position on the DNA strand, on the codon, because that's the wobble position. So your AGT became AGC, but guess what? Even though this is a different codon, UCG is not the same as UCA, but they both code for serine. Remember, because of the degeneracy of the genetic code. The genetic code is degenerate, so there's more than one codon for a particular amino acid, and that works out great here. So here's just a rundown of the different types of mutations and their definitions for you to, take a look at. I think the only one I left out, I really didn't touch on suppressor mutations or intragenic suppressor or intergenic suppressor, but we pretty much touched on the rest of these. So take a look at this and learn, each one of these are important and good to know. So let's do concept check number 2 and then take a quick break with Gizmo and Wicket and then come back strong to finish this chapter. Let's do that. So concept check number 2. How does a suppressor mutate? Oh, I just told you I'm not gonna ask you about suppressor mutations. So in fact, let's just go ahead then and do our break and we'll come back strong to finish this chapter. What do you say? All right, welcome back from break time with Gizmo and Wicket. Let's go ahead and finish off this chapter. So one thing you should be aware of is the term mutation rates. Mutation rate refers to the frequency at which a wild-type allele at a locus changes to a mutant allele. And this could be expressed as rates per replication, rates per spore formation, rates per gamete. Okay, and we can see here that overall the rates are actually quite low. So you've got 10 to the minus 5 to 10 to the minus 8 occurrences. So overall, your DNA replication machinery does a great job at preventing mistakes, right? So when DNA polymerase goes ahead and copies the DNA during cell division, there is a chance that it can make a mistake and maybe put a T where it should have put a C, for instance, and that does happen. But remember, we may have touched on in a previous chapter proofreading ability of DNA polymerase. DNA polymerase has what's known as proofreading ability. It has a 3' to 5' exonuclease activity, which means it's chugging along copying the DNA. If it puts a C instead of an A, it could catch that, it could realize that, and then back up. Remember, normally a DNA polymerase travels 5' to 3'. Well, if it puts down the wrong nucleotide, it can actually stop, back up 3 to 5, back up, cut out the nucleotide. That's what exonuclease activity means. Cut out the improper nucleotide and replace it with the correct nucleotide. So DNA polymerases have proofreading ability. They have the ability to check their work. And because of that, they make remarkably few errors. Now, have you ever heard of, let me share something with you that you might find interesting. Have you ever heard of the flu virus, mutating frequently? Or have you heard the fact that COVID, they're worried about COVID replicating too quickly? It's a coronavirus, and they're worried that it's gonna not replicate too quickly, but mutate too quickly, and the flu mutates too quickly. And this is why you need seasonal flu shots, and you may in the future need seasonal COVID shots as well. You ever wondered why the SARS-CoV-2 virus or the flu virus mutates so quickly if what we're seeing here is remarkably low rates of mutation? Do you know the answer to that? Here's the answer. Those viruses are RNA viruses. Remember, I told you that viruses can have RNA genomes? Well, the flu virus has an RNA genome, and also SARS-CoV-2, the virus responsible for COVID, is also an RNA genome virus. RNA polymerase does not have proofreading ability. RNA polymerase does not have proofreading or exonuclease activity. It can't check its work. It makes mistakes all the time. It doesn't care, right? So RNA viruses are always going to give you higher rates of mutation compared to DNA viruses or any of the DNA organisms because of that property. So now you know something interesting. Now you know why the flu virus mutates so quickly and why scientists are concerned that SARS-CoV-2 is going to mutate so quickly. It's an RNA virus, and RNA polymerase can't check its work. And if you're not checking your work, you make mistakes. Interesting stuff, right? So let's carry on with this. So how are mutations caused, right? How do you get a mutation? Well, some mutations can be spontaneous. This would be an internal factor. So it just spontaneously happened. I don't know, your DNA polymerase could make a mistake and not check its work properly and just make just a silly mistake. So these occur under normal conditions. And we're going to talk about some examples of spontaneous mutations. Then you can have induced mutations. These are caused by environmental chemicals or radiation. So, when they say don't smoke, you'll get cancer. This is what they're talking about. You're inhaling chemicals that can cause DNA damage, that can cause mutation and cancer. So let's talk a little bit about the spontaneous errors, the spontaneous mutations. So you could have mispairing due to other structures. So what does this mean? Let's look at figure 310 a little bit. But normal, protonated, and other forms of the bases are able to pair due to wobble flexibility of DNA's helical structure. So this is a problem with the wobble effect, but not in a codon, but in DNA itself and DNA's flexibility, itself. So take a look here. Sometimes T's can pair with G's just because of a wobble effect, because of just the inherent flexibility of the DNA helix itself. And also protonated adenine can sometimes pair up with cytosine. So you can see these are problems due to wobble effect that can affect base pairing and can cause spontaneous mutations. And those spontaneous mutations that are due to wobble can then become stably incorporated into the genome. Let's take a look at that. So here we have our double-strand DNA. It's wild type. The strands separate for DNA replication. But see, thymine on the original template strand pairs with guanine through the wobble effect, leading to an incorporation of an error. So instead of thymine pairing with adenine, thymine pairs with guanine, right? Because DNA polymerase made a mistake due to wobble, and now that's going to be inherited. That's going to be stably inherited from then on, and now it's a mutant. It's going to be a mutant cell, a mutant allele, that is. So at the next round of replication, DNA replication, the guanine nucleotide pairs with cytosine, leading to a transition mutation. Transition because it went from a pyrimidine T to a purine G, a transition mutation. And then there are spontaneous causes of deletions and insertions as well. So you can end up with a frame shift as well due to spontaneous reasons. This is either caused by strand slippage or unequal crossing over. So in strand slippage, you have one nucleotide strand forming a small loop, and that can cause either introduction of a nucleotide or deletion of a nucleotide, so a frame shift, or unequal crossing over, which results in one molecule with an insertion, the other with a deletion. So let's take a look at these. Here is an example of insertion and deletion due to strand slippage. So here you see you've got your template strand, and then the newly synthesized strand loops out, you see, so you have strand slippage. Instead of AAA, one of the A's has looped out, and because of that, when the DNA is copied, resulting in addition of one nucleotide in the new strand when the DNA is copied. So now you have there's only addition of one nucleotide. On this new strand. So you have this strand has an extra A because it looped out and it wasn't recognized. Here, the template strand loops out, the template itself loops out, and that results in the omission of that nucleotide on the new strand. So here you end up with an extra adenine, or adenosine, I should say. Here you end up missing an adenosine. So you see, due to strand slippage, you can either gain a nucleotide if it happens on the daughter strand, if the loop out happens on the daughter strand, or lose a nucleotide, and that is if the loop out happens on the template strand. And then remember, you can also get insertions or deletions due to uneven crossing over. So you see, these are homologous chromosomes. This would be a homologue of this one, so you should have crossing over that happens between the two sequences, but let's say crossing over happens at the end of this sequence, so not directly in between the two. Well, now you could end up with too much information on this chromosome, so that would be an insertion on this chromosome, and too few nucleotides on this chromosome, so that would be a deletion on this chromosome. So another example of just spontaneous mutation there. Now again, you can have spontaneous replication errors. You could have spontaneous chemical errors too. Okay, we're going to talk about that in just a second. Chemical changes that are spontaneous. And then we'll talk later on about the, smoking and whatnot, chemically induced mutations or radiations. But what are spontaneous chemical changes? So spontaneous chemical changes could include depurination, which is the loss of a purine, deamination, which is the loss of an amino group, the amino group on the nitrogenous base. Take a look here. Here's depurination. Occurs when the covalent bond connecting a purine to the 1' carbon is broken, indicated by the dotted line. You see? So just spontaneously, this covalent bond is broken, and the purine guanine floats off. Now it's naked. There's no, it's just sugar phosphate. There's no nitrogenous base anymore attached to that purine. Okay, and then that would lead to a apurinic nucleotide. So, and then replication of a template strand with an apurinic site may lead to incorporation error. So see what happened? You had your DNA, it got depurination. You lost that G, remember? The covalent bond was cleaved. G is gone. Once you have strand separation, a nucleotide with the incorrect base, in this case A, most often A, is incorporated into the newly synthesized strand. You have strand separation again. At the next round of replication, this incorrectly incorporated base will be used as a template. So now you have a permanent mutation due to originally what was depurination. And then number 5, a nucleotide is incorporated into the newly synthesized strand opposite the apurinic site as well. So you end up with mutations that are permanent and stable. And again, this was spontaneous. This can happen spontaneously. It's just bad luck, right? And that's why it's impossible to say, for instance, that you're going to avoid all mutations, right? So, people say, could you still get cancer if you avoid every possible bad thing? Like you avoid going out in the sun, you avoid x-rays, you avoid smoking, you avoid drinking, you avoid all the bad things in the world. Could you still get cancer? And the answer is yes, it is still possible, much less possible, much less likely than if you smoke or drink or do all these other things. But if you just lead a life that's completely avoidant of all of these issues, all of these chemicals and carcinogens and radiation, you could still end up with cancer, unfortunately. One of two ways. One is you inherited an already mutated gene that's heritable cancer, or two, just one of these spontaneous events happened, you know, and that could lead to mutation and cancer. Interesting, right? So deamination, that was the other one. Remember I said deamination is the loss of an amino group? Let's look at that one. So deamination, you have cytosine. It becomes deaminated, spontaneously deaminated. So the amino group goes away. Now cytosine gets converted to uracil. That's not good. So now you have, you know, an error here. And what pairs with uracil? A's pair with U's. Instead of a G pairing with this C, now an A will want to pair with this U. Okay. Similarly, you could deaminate cytosine at a different site and end up with thymine as well, right? So you could end up with thymine, you could end up with uracil. It all depends. All right, so now this one is what people think about mostly when they're thinking about mutations. They're thinking about, well, you incurred a mutation due to a mutagen or radiation or something. That's called a chemically induced or radiation induced mutation. So let's start with chemically induced. Chemically induced mutations are usually caused by what's called a mutagen, any environmental agent that significantly increases the rate of mutation above the spontaneous rate. Mutagen, anything that causes mutations is a mutagen, right? So, for example, here's an example, base analogs, a class of chemical mutagens with structures that are similar to your 4 standard bases, nucleotide bases of DNA. So it looks like an A, G, C, or T, but it is not. It's an imposter, right? It's an imposter chemical. And one of the common ones here is 5-bromouracil, which kind of looks like thymine, right? So let's read here. 5-bromouracil, a base analog and a mutagen, resembles thymine, except that it has a bromine atom in place of a methyl group on the 5-carbon atom. Because of the similarity in the structures, the 5-bromouracil may be incorporated into the DNA on accident in place of thymine, so instead of a thymine, and then like thymine, 5-bromouracil normally pairs with adenine, but when ionized, it may pair with G's, guanine, through wobble instead. Could you see how that would be a problem? So normally 5-bromouracil will pair with adenine, which is okay. But when ionized, see the negative charge here? When ionized, it might bind to guanine, and that could induce a missense mutation. What other mutagens are there? Well, there could be alkylating agents, deamination enzymes, or hydroxylamine. That's another example of a mutagen. These are all mutagens. So let's talk about alkylating agents. They donate an alkyl group, like a methyl group or something. So let's take a look here at an example of each. What does an alkylating agent do? What does deaminase do? What does hydroxylamine do? So you see the original base here is guanine or cytosine. Guanine undergoes alkylation. And you see it gained a methyl group here. Or actually, or is that an ethyl group? It gained a ethyl group, and then that allowed it to pair with thymine. You see, because of its alkylation, it now pairs with thymine, where it should pair with what? Cytosine. And then you get a mutation, a missense mutation, right? With deamination, you see you remove this amino group on cytosine. Now it pairs with adenine, where it shouldn't pair with adenine. It should pair with guanine, and that leads to a missense mutation. Now you have, here's an example of what hydroxylamine does, which is called hydroxylation. So here you add a hydroxyl group to cytosine, and now it pairs with adenine. It should not pair with adenine. It should pair with guanine. So you can see by chemically modifying these nitrogenous bases, you're causing mispairing, and that can result in mutations, missense mutations. Okay. Another group of mutagens are called intercalating agents. Intercalating agents, what they do is they kind of get into the nitrogenous bases. They intercalate into the DNA. So they kind of embed themselves in the DNA. They sandwich themselves in between the bases of the DNA. So they're molecules that love to sandwich themselves. You see these little rectangular structures. They intercalate into the molecule, into the DNA. And when that happens, that can cause deletions or insertions during DNA replication. And you may have heard of, you probably haven't heard of these ones, proflavin or acridine, orange. You probably haven't heard of these, but if you have ever done any work in a molecular biology lab, you may have worked with ethidium bromide. That's the chemical you add after you've done PCR. You know, we'll talk about this stuff in the next chapter actually, but after you've done PCR in the molecular biology lab, you want to see your DNA under UV light, so you add ethidium bromide. Well, ethidium bromide is an intercalating agent, and that's why when you're working in the lab, you've got to wear gloves and you have to be really careful around the ethidium bromide, because the molecule intercalates into the DNA, and that can cause insertions and deletions. So concept check number 3, base analogs are mutagenic because of which characteristic? Why are base analogs mutagenic? They produce changes in the DNA polymerase? Nope. They distort DNA structure? Nope. They are similar in structure to normal bases. That's exactly right. They look like normal bases, so they get incorporated as such by DNA polymerase, and then that results in a mutation. All right, now lastly, let's talk about radiation and what goes on with radiation. It's another type of induced mutation. So we're talking about things like UV, X-ray, you know, there's other types of radiation as well, gamma radiation, but we're talking UV and X-ray mainly. X-rays, by the way, X-rays cause double-strand breaks in the DNA. I don't think I wrote that here. X-rays cause double-strand DNA breaks. A UV causes what is known as pyrimidine dimers. So when you have UV light, if you have two pyrimidines, they can dimerize. So thymine, if you have two thymines, they can form a covalent bond and form what's called a thymine-thymine dimer. If you have two cytosines, it can form a cytosine-cytosine dimer. If you have a cytosine and a thymine, it can form a cytosine-thymine dimer. Now, thymine-thymine dimers are the most common ones, but you can have the other ones as well. And usually thymine-thymine dimers lead to cell death, right? This is cell death. This is one of the main reasons you go out in the sun for too long and get a sunburn and then all your skin dies. You know, that's one of the main reasons, is because you got all these thymine-thymine dimers, and you actually got a lot of damage to your cells and to your DNA as well. This is why it could lead to mutation and lead to skin cancer that way. Now, in bacteria, you have what's known as the SOS system, which allows the bacteria to bypass the death, the replication block or death caused by these pyrimidine dimers, but it's a mutation-prone pathway at the expense of incurring a lot of mutations. Some bacteria have this SOS system that allow them to persist in the face of UV. Especially there's bacteria called Deinococcus, and those ones can withstand a lot of UV damage. And so with the help of SOS system, they're able to persist and keep growing, but they incur a lot of mutations in the process. So now you can see what happens. You have two thymines in a row on your DNA, two thymines. You go out in the sun too long, and the thymines, dimerize. They form covalent bonds between them, you see? And on the DNA, this causes a bulge out on the DNA. You see a distorted DNA molecule bulging out. Now, what happens when you have these problems? Well, there's a number of ways to fix the problems. That your body has a number of different ways to fix damaged DNA. Okay, and bacteria as well. Life has a way of repairing DNA damage. First of all, you could prevent the DNA damage from happening to begin with. You have proofreading, remember 3' to 5' exonuclease activity, proofreading of DNA polymerase. But let's say something escapes the proofreading or something bad happens and you end up with mismatch. Well, you have a mismatch repair mechanism which corrects incorrectly inserted nucleotides using enzymes. You have specialized repair enzymes in your cells. Direct repair, this would be you're restoring altered nucleotides to their correct structure. So you remember how deaminases would like deaminate nucleotide? Well, an enzyme could come back and add the amino group back on, for instance, directly repairing that nucleotide that was damaged. Base-excision repair, so you would remove a bad nucleotide or an incorrect nucleotide and replace it with the correct one. Nucleotide-excision repair removes bulky DNA like the thymine-thymine dimers, for instance. So you could actually go in, there are enzymes that'll go in, remove the entire thymine-thymine dimer and then replace it with proper thymines. Here's just an example of the mismatch repair. Remember the mismatch repair mechanism. So you've got your double-strand DNA. Let's start here at the top. In DNA replication, a mismatch base was added to the new strand. So this is the new daughter strand of DNA. Number 2, methylation at the GATC sequences allows old and newly synthesized nucleotide strands to be differentiated. A lag in methylation means that immediately after replication, the old strand will be methylated and new strand will not. So this is something we haven't touched on yet, but I should mention it now. When the DNA inside of your cells is methylated at these GATC sequences. You have methylated DNA at the GATC sequences inside of your cells. And your DNA repair machinery knows to scan for these methylated sequences because only your template strands have the methylation. Once DNA is copied, the daughter strand will not have those methyl groups on the DNA. They only incur those methylations afterwards, after DNA replication is finished. The daughter strand will become methylated as well, if that makes any sense. So your DNA repair machinery needs to know, well, what's the old strand and what's the new strand? Because I got to repair the new strand. I don't want to repair the old strand. It looks for these methyl groups, and when it sees a methyl group, it says, oh, that must be the old strand. I'm going to use that as the template to repair the new strand of DNA. Not something you really need to know, but it's interesting to know. So let's move on. So you found out where's the methyl group. So this bottom strand is the template strand. It's the correct strand. So that means that this top strand is the incorrect strand. It's the daughter strand with the information that needs to be fixed. So then these proteins attach, the mismatch repair complex. These proteins attach and bring the mismatch bases close to the methylated GATC sequence, and the new strand is identified. It says, oh, okay, look at this. The new strand is identified. This G is wrong. Then an exonuclease removes the nucleotides on the new strand. See, it removed all these nucleotides from the new strand. And then a DNA polymerase then fills in all that information with correct information. And you've now repaired the misplaced this G with the correct A. And you've fixed the problem. And here you can see a methyltransferase repairing. Remember direct repair I told you about? You could directly repair a molecule. So here's a molecule that has been damaged, right? It's been modified into O6-methylguanine because it has this weird methyl group here. Well, a methyltransferase can come here and directly repair this back to guanine by removing that methyl group. Does that make sense? Awesome. So really interesting chapter. Again, this is why life persists is mutations and genetic variation. So it's so important to life, but at the same time, it really does bring suffering to organisms in that you can incur mutations and get cancer. You can incur mutations and pass them on to your offspring as genetically inherited disorders. but it's fascinating to understand and learn about. So again, as always, it's been a pleasure. Let me know in the comment box below if you have any questions, and I'll catch you guys next time.
Dr. D, Dr. D, Dr. D. 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 Dr. D, Dr. D, Dr. D.